Students, in this class, we are going to learn some of the important diseases which are all causing economic yield loss in grapes, which includes downy mildew caused by Plasmopara viticola, and then powdery mildew caused by Erysipeia necator. Earlier, it was known as Uncinula necator, and then bird's eye spot or the anthracnose caused by Alcino amphelina, and then black rot caused by Gugnardia pudwicki, rust caused by Phacospora vitis or U vitis, and then brown leaf spot caused by Cercospora viticola, and then some of the leaf blight caused by Alternaria, and mold caused by Botrytis, etc., and by the soft rot bacterial canker caused by soft rot by Phomopsis, and then bacterial canker by Xanthomonas. We are the important diseases, and we are going to see these diseases and the management practices one by one in the subsequent slides. And besides the field infection, the fruit rots, which are mediated by several pathogens, are also very much important, which are all caused by first Losiapsis or Penicillium or Rhizopus or Cladosporium, Grignardia, and then Pseudomonas, etc., are also responsible for several fruit rot symptoms, especially on the grapes. And first we are going to learn about the downy mildew, which is first reported in Europe during 1878 and have the epidemic in the France during 70 to 80 and leads to 50 to 100% yield loss, complete defoliation of the affected plants. And and this is having the historical background that the historical wonder chemical Bordeaux mixture have been developed during 1885 by Pierre Alexis Milladet, P.A. Milladet, for the management of this downy mildew, especially to manage this downy mildew on grapes in France from the area Bordeaux, and hence it is named as Bordeaux mixture. And all this pathogen is known to affect all the aerial or tender parts of the plants. Upon the leaf on the upper surface, it produces yellow chlorotic spots, and the down surface of the and corresponding to the under surface, you could see the downy growth or white growth. Downy means velvety growth. This white powdery growth that represents the sporangiophore and sporangia of the fungi, and the severe infection that may lead to necrosis of affected area, and which turn into dark brown and ultimately results in premature defoliation. And a similar type of downy growth or patchy dull downy growth may also be appear on the tender shoots, which are turned into dark brown in color with cottony growth. Upon the fruits, the berries which turn hard and leathery, and the affected berries are shriveled, dried, and which is mentioned as mummified fruit, and which shells and the pulp inside the fruit is also been affected. And the pathogen responsible for this downy mildew is Plasmopara viticola, and which produces sporangiophores. These sporangiophore emerge out through the stomata as well as through the lenticels. The infected leaves as well as the vines, upon the infected leaves and vines, the pathogen known to produce the sexual spore, that is oospores, they remain dormant for a longer period. Besides, the dormant mycelium present in the infected plant debris are also responsible for development of this symptom. And this is the typical white downy growth on the undersurface of the leaf. Corresponding to the upper surface, you could see the yellow discoloration, and which is the indication that the leaves are infected with downy mildew symptoms. And the fruits also, the rachis got affected due to severe infection, and thus that may bend, giving a S-shaped structure and completely turn into reddish brown in color due to infection by the downy mildew pathogen. This is the downy growth that found on the young inflorescence as well as on the berries, and the affected berries are become completely wrinkled, shriveled, and internal pulp got affected, and which is become mummified. And this is the typical symptom by which you could differentiate the powdery mildew as well as downy mildew infection at the field level. and the life cycle that includes the presence of the oily spot as well as the fruit infection. The pathogen germinate and enter inside the tissues and establishes the host area through which it absorbs the nutrients, and through the stomata the sporangiophore that emerge out here. The sporangiophore is of very typical, which is right branched right angle, that is right angle branching is the typical character of Plasmopara verticala. And upon these right angle branches, upon the the sterigmatas are formed, which bears a single oval shaped sporangia. These sporangium are dispersed by the rainwater, and the internal contents are cleaved into formation of zoospores, and these zoospores are released into the environment. They swim in the thin film of water for some time, sheds its flagella, become encysted, germinates, and establishes the infection. By the way, the life cycle repeats. The another way, from the sporangia, from the mycelia, it produces antheridium as well as the oogonium, which is responsible for the production of oospores through sexual reproduction. These oospores, upon the favorable condition, they germinate and produce the sporangium, and the internal content of these sporangium are cleaved into zoospores, which swims in the what is zoospore? The zoospores are kidney shaped or reniform, biflagellate, anteriorly tinsel and posteriorly whiplash type of flagella. And this zoospore swim in the thin film of water with the help of flagella, and sheds its flagella, become encysted, germinate, and establishes the infection. By the way, the life cycle that is asexual as well as sexual life cycle that repeats, and it survives on the infected. This is the typical right angle branching. This is the typical right angle branching of sporangiophore, which bears a single spherical shaped hyaline sporangium. And the pathogen that survives in the infected leaves as well as the vines as oospore as well as the dormant mycelium. So the infection, primary infection that starts from the oospore as well as the dormant mycelium present in the infected plant debris, while the secondary spread is mainly through windborne sporangia and zoospores. The temperature range of 20 to 23 degrees Celsius with the relative humidity of 80% helpful in serious dissemination of this disease, are favorable for the development of this disease. And to manage this disease, remove completely destroy those affected plant parts, and planting of vines with the proper spacing and periodical training or pruning should be done to avoid spreading of the disease. Spraying with the podo mixture at the rate of 1% or difolaton or chlorothalonil or metalaxyl at 0.2%, and it should be repeated at weekly intervals or 10 days, or the interval must be decided based on the intensity of the disease. And there is a biocontrol agent, that is Erwinia herbicola, which is a saprophytic bacterium, which may also be used in the liquid culture, which reduces the population of which inhibits the Plasmopara verticala up to 75%, and this is still under research, and it is not commercially available. And next one is the powdery mildew, which is caused by Uncinula necator, and it is renamed as Erysipeae necator. So the Erysipeae, which is known, which is the indication that it is a oidum type of powdery mildew, which produces white powdery patches, especially on the upper surface of the leaves. And this is more serious than that of the downy mildew infection. The infected berries become more acidic and not suitable for brewing industries. So infection on the berries, that is powdery growth on the berries, that causes cracks on the outer rind of berries and invites several airborne pathogens as well as saprophytes. It was first reported from England, and now it is prevalent throughout India and everywhere. This pathogen is also known to affect all parts of the plants. So the infection on berries that lead to death of epidermal cells, and their growth is prevented as the internal pulp continues to grow and develops the skin cracking, and this crack invades the infection. and berries become misshapen and irregular in shape, and lesions on the leaf which also turn into necrotic, and when the pathogen covers the entire leaf ultimately leads to drying and defoliation. Upon the defoliated leaves, the sexual Ascocarp chasmodium will be produced, which in turn contains ascus and ascospores. So these ascospores are responsible for the primary spread, and an airborne conidia is responsible for the secondary spread. And this disease will be more severe during the October to February, when the warm dry weather or warm humid weather prevails. This is the typical powdery patches on the upper surface of the leaves, and this is the typical symptom that formed on the inflorescence or on the young berries, ultimately leads to malformation or wrinkling of leaves, ultimately leads to drying and defoliation. This is the powdery growth on the berries. Similar type of powdery growth can also be seen due to downy mildew infection. How to differentiate? In the case of downy mildew infection, the affected portions become mummified, while in the case of powdery mildew infection, the affected berries may develop cracks on the outer rind, and the internal pulp got exposed due to the infection by the powdery mildew pathogen Erythema necator. And this is the idiom type, as we know, which produces the short conidiophore, which bears barrel shaped conidia arranged in a basipetal succession, and during the sexual stage it produces chasmothesia, which remains in the infected or defoliated leaves, which contains ascus and ascospores responsible for the primary spread. And this leaf typically shows the downy mildew symptom, that is yellowing on the upper surface, while the powdery mildew that produces white powdery patches on the upper surface. In the case of downy mildew, yellowing will be on the upper surface, and corresponding to the under surface you could see the white downy growth. And this is the life cycle of the pathogen, where it produces the spores and spreads through the air, establishes the parasitic relationship with the host plant, and overwinters in the affected or infected plant debris or the buds, and finally it spreads to the new flushes. It survives as like that of other pathogen, other powdery mildew pathogen. Here also it survives as a dormant mycelium as well as chasmothesia on the fallen leaves as well as on the infected shoots, and the ascospore from this infected portion serves as the primary source of inoculum. Airborne conidia responsible for the secondary spread, and this disease is more severe during the October-November in northern part of India, while it is more severe in from February to June in southern part of India. And this disease is favored by warm, humid weather and the sultry weather, which is responsible for high incidence. And the warm winter temperature of 20 to 33 degrees Celsius favors the development of this disease. So avoid overcrowding or dense growth of vines, so that it should be which could be achieved by periodical pruning of the grapes wine is recommended to give sufficient air circulation. And then when the infection is noticed, then go for spraying with the wettable sulfur at the rate of 0.2%, or dinocap 0.2%, or carbendazim 0.1%, or the latest generation fungicides like azoxystrobin, trifloxystrobin, etc. can also be used. And in the case of biocontrol, you can go for application of Bacillus subtilis, or the mycoparasitic fungi Ampelomyces quisqualis are also recommended to manage the disease under field level. The next important disease is anthracnose or bird's eye spot, which is caused by Elsina ampelina, and the perfect stage is Phaseoloma ampelinum. And the shoots, tendrils, petiole, leaf, vines, stem, inflorescence, berries, etc. All the parts of the plants got affected by this Elsina ampelina, and upon the leaf it produces small, irregular, dark brown spots with grey center. And in severe case of infection, the center portion may fall off and leaving a shut hole symptoms. And the similar type of deep-seated spots will be formed on the petioles as well as on the leaves, and the affected leaves will be distorted. And the spots, several such spots unite together and forming a deep-seated lesions on the stem ultimately leads to development of dieback symptom. And upon the berries also, it produces a characteristic round spot with the dark margin and sunken spots with the gray center, which resembles the bird's eye. That's why it is mentioned, nicknamed as bird's eye spot. During the asexual reproduction, it produces the acerulae at the center of the gray spots, which in turn contains numerous conidia, conidiophore, and setae. Here the conidia, which are single-celled, hyaline, oblong or ovoid in nature, while during the sexual reproduction, it produces pseudothecia, which in turn contains ascus and ascospores. This is the typical symptom that formed on the surface of the leaf. Deep-seated lesions formed on the vines or the stem. This is the infection or sunken black spots on the berries. Later got extended with the reddish brown margin and grey center. And this one that resembles the bird's eye, and hence it is nicknamed as bird's eye spot, and which become deep-seated. See here, this picture will shows the typical bird's eye like appearance. This is the deep-seated infections on the vines, and the portions that are just above the point of infection are completely dried, and the leaves become yellow and dried, leading to dieback, development of dieback symptom. This is the severe infection on the bunch of berries. And the pathogen that survives as the dormant mycelium on the cankers or on the stems as well as on the twigs. Here the conidia are single-celled oblong, as I told earlier, and this is carried through the wind and responsible for the secondary spread. And this is very severe during July to August and November to December. Infection on the new sprout that occurs during the rainy season. So rains after the pruning, which is responsible for severe incidents. And to manage this disease, remove out the affected portions and completely destroy, and use some of the resistant varieties like Anchor Call and Bangalore Blue, Beauty Seedless, etc., which are mentioned in this slide, can be explored as a resistant cultivars. And if the disease is noticed in the existing plant, then go for application of Bodo mixture, 1% Bodo mixture, or copper oxychloride at 0.25%, carbendazim at 0.1%, or mangozeb, typhloton or chlorothalonil at the rate of 0.2%, which are effective against the development of this disease. And then the black rot disease, which is caused by Guignardia pardwicki, which produces red necrotic circular spots with a dark black margin, and such spots are formed on the leaves. At the center there will be a black dots, which represents the pycnidia of the fungi. Similar such depressed black lesions that are all formed on shoot, tendrils, petioles, flower stalks, etc. Upon the fruits or berries also, it produces discoloration, spots, and ultimately leads to decaying of affected berries. The whole berry become affected, rot, shrunken, and turn into dark black in color, and hence it is mentioned as black rot. This is the typical brown spots that produced on the surface of the leaf and later get extended with the dark black pycnidial bodies at the center of affected leaf portions. In this portion we could see the presence of pycnidia. And the affected berries turn into dark black in color, and and hard in nature, which could be easily differentiated from the infection by the downy mildew pathogen. And here in the completely shrunken and blackened berries, and the pathogen known to produce the sexual ascocarp called perithecia. So the ascospores from the perithecium that serves as primary source of inoculum, while the pycnidiospores from the pycnidium, which serves as the secondary source of inoculum. And for managing this disease, as we studied in the many other diseases, collection and destruction of affected plant portion that reduces much of inoculum potential. And in the ongoing crops, you can apply Bodo mixture at the rate of 1%, Captan or chlorothalonil at the rate of 0.2%. They are found to be effective in reducing the disease incidence. And this is the life cycle. So upon the mummified fruits, The perithecia are developed in the perithecia, which in turn contains ascus and ascospores. This is the ascus with the ascospores, and these ascospores fall on the leaf and develop the lesions on the leaf as well as on the young fruits. Upon the affected portions, the pathogen is known to produce the pycnidia, and pycnidia in turn contains numerous conidiophore and small single-celled hyaline conidia or pycnidiospores, and these pycnidiospores carried through the wind falls on the bunches as well as on the leaves, where it causes the infection. This is the asexual reproductive cycle, and this is the sexual reproductive cycle of the pathogen. And the next important disease is the rust disease, which is caused by Phacopsora vitis or Phacopsora uvitis, and it is very severe in southern part of India, and black prince. The variety black prince is highly susceptible, and winter season the incidence will be much high during the winter season. It is a heterogeneous rust. What does it mean? It definitely needs another host to complete its life cycle, and hence the spermagonial as well as the acial stages that are all formed on the alternate host called Meliosma meriantha, while the uridial, teleal and basidial stage that occurs on the target host, that is on the grapes. This is the grape leaf infected by the Phacopsora uvitis and developed uridospores, and this is the Meliosma meriantha, which serves as the alternate host, and which is highly required to complete its life cycle. Initially, it produces yellow to brownish lesion on the upper surface, and corresponding to the under surface, you could see numerous orange yellow color raised pustules. Such pustules which will cover the entire leaf and ultimately leads to drying and severe defoliation due to severity of this disease. And this pathogen Phacopsora uvitis is known to produce single-celled iridospores. These iridospores are binucleate, hyaline in nature or with some colored walls, and accenulated, pedicellated and accenulated. Here, while the teleospores, which are sessile in nature with a smooth outer wall, here also single-celled with one germ pore, and it is dark brown in color. This is the typical discoloration on the upper portion of the leaf, and corresponding to the under surface it produces orange yellow color pustules, and these orange yellow color rust pustules which turn into dark brown in color at the time of maturation when the teleospore are formed. And this is the these plants are showing the varied intensity of disease incidence, and this is the experimental plant which was inoculated with the Phacopsora uvitis and shows this heavy infection on the leaves of inoculated plants. When the disease is very severe, then go for spraying with the Zinap at the rate of 0.2%, or spraying with the wettable sulfur at the rate of 0.2%, dusting with the sulfur at the rate of 25 kg per hectare, or spraying with Vutavax at the rate of 0.1%. All are found effective in reducing the disease incidence. Even you can go for application of recently released broad spectrum fungicides coming under the group Strobilurins, which will also helpful in reducing the incidence. And then the bacterial canker, which is caused by Xanthomonas campestris pathova viticola, is also a serious disease on grapevine. Initially, it produces water-soaked lesions surrounded by the yellow halo on the lower leaves, lower surface of the leaf. Later, these spots become enlarged, necrotic, turn into dark brown in color. As I mentioned, in many year crops, this bacterial infection, especially on the leaves, that the symptom development or the infected area is restricted by the veins and veinlets, and hence it has become more angular in nature, and it form large patches on the infected leaves. And when the infection is very severe, the bacteria enter in the veins of leaves and also causes venal necrosis. Besides, it also produces some cankerous growth on the petiole or on the stem as well as on the berries. The pathogen Xanthomonas campestris pathova viticola, which is a gram-negative rod-shaped bacteria with a single polar flagella, and the bacteria may survive on the alternate host like neem, mango, etc. Wind as well as the rain splash are responsible for the spread of this bacteria. And to manage this disease, you should collect the cuttings from disease free area. And during the cropping period, if you notice the infection seriousness of this infection, then go for application of streptocycline at the rate of 300 ppm and repeat at 15 days interval based on the intensity of this disease. This is the leaf necrosis symptom due to bacterial infection, and here you could see the bacterial cankers formed on the stem due to severity of the bacterial infection. And this is the bacterial colonies which were isolated under artificial conditions. And some of the leaf spots which are also caused by cercospora as well as the alternaria. And how to differentiate these leaf spots on the surface of the leaf? The cercospora spots will be with the dark brown margin and somewhat light brown or gray center, while the alternaria spots initiates as a dark brown, later get enlarged and produces concentric circles on the affected area. By the way, you could differentiate the leaf spot caused by cercospora viticola as well as leaf spot caused by alternaria vitis. In both the cases, several severe, when the disease is severe, several such spots coalesce together and cause complete drying and defoliation of the leaves. And when these type of spots are appeared on the plant, then go for this can be controlled or managed by application of Mancozeb at the rate of 0.25% or application of Bordeaux mixture at the rate of 1% are quite effective. And some of the varieties like Australia number 2, Champak and Champion, which are found to be resistant to these sort of leaf spots. And the Phomopsis is also known to cause some of the leaf spots as well as cracks and lesions on the vines of grapes. And in the longitudinal section you could see the internal discolorations which are also quite common, and the berries are also which much reduced in size, and this may completely be dried when the intensity will be much more. And this Phomopsis blight is caused by Phomopsis viticola and botrytis. This botrytis, which is known to produce grey mold on the affected berries especially, and this botrytis is also known to cause some of the leafy infection. And the botrytis infection which will be much higher during the cool period, that is winter season, and hence this botrytis mold is a serious problem in temperate countries. And it is known to produce symptoms on shoots, flowers and berries, and it produces irregular necrotic spots at the center of the leaves, and marginal necrosis induces the marginal necrosis, and the infected flowers are also become completely necrotic and covered with a tuft of sporulating mycelium. When the berries are affected, and the affected berries are completely covered with grey mycelial mat with profused conidial growth. The prominent symptoms are found especially on the berries. And then, as I mentioned, the infected berries become dark colored, typical greyish hair-like mycelia and profused sporulation. The tuft of conidiophores as well as the conidia, which are protrude from the stromata formed on the skins of berries. Under high disease pressure, all the berries in the bunch are affected by the botrytis cinerea. This is the typical symptom developed by botrytis cinerea and leading to bunch rot. And this is the profused mycelial growth and sporulations on the infected plant, infected berries. Here also you could see the formation of lesions, the initiation of the lesions and final sporulation on the affected berries. And this pathogen which survives on the grapevines as well as in the rotten berries or as cluster of mycelium as well as sclerotia. So the sclerotia present in the infected plant debris serves as the primary source of inoculum, while The pathogen known to produce bunch of grapes bunch like conidia and the verticillate conidiophore, and these conidia are responsible for the secondary spread. And the low temperature obviously favors the development of the disease, and the temperature range of 20 to 25 degree with high relative humidity are responsible for germination of this sclerotia, and upon germination of the sclerotia it is known to produce apothecium, which in turn contains ascospores, and these ascospores responsible for the initial spread, and the conidia developed from the mycelia, which is responsible for the secondary spread. Sanitation of the vineyard is the important cultural practice to reduce the inoculum density in the field. The diseased vines, leaves, and fruits must be completely collected and destroyed. Spraying of proteone or the triazole or azole group of fungicides are recommended to reduce the incidence under field level. And the next one is the crown gall, which is caused by the bacterium Agrobacterium tumefaciens. The infection by the bacteria that is responsible for developing a galls at the site of infection. And this disease was first found, and the next one, sorry, at the base of affected plants near the soil level, and this gall initially that would be white, pulpy, and soft, gall like profused growth. Later that would become hard, irregular in shape, and due to development of these galls, the transportation of water and nutrients from the base to the upper part of the plants are completely cut off, ultimately results in drying and death of affected plants is most common in the case of grapevine. And then grapevine fan leaf disease, which is a viral disease which is very predominant, and it was first reported in California during 1950, and in India it is reported in 1965. So the infection produces sharply toothed leaf margins and mottling on the young leaves, closeness of primary vines, and then yellow mosaic pattern on the leaves, and thickening and vein banding, and malformation of the affected berries. All are the typical symptoms. Here the yellowing and stunted growth of affected plants, all are the typical symptoms caused by the grapevine fan leaf virus. So the affected vines are stunted in growth, less bigger than the normal one. The leaves are severely distorted and become asymmetrical, cupped, puckered, and exhibit acute dentation. That is dents, like what I can say. Uh, it's a trough like structure, and the chlorotic mottling sometimes that accompany with the all foliar deformations. The vines are also become malformed, abnormal branching and shortening of internodes are the common symptoms, and the bunches are reduced in number as well as size, and ripening process ripen irregularly and have short berries or poor berry setting. The pathogen responsible for this disease is the grapevine fan leaf virus, which is an epovirus having isohedral shaped RNA virus, got transmitted by the dagger nematode Ziphinema index. And better manage this disease with the nematicides or removal of affected plants, which will helpful in spreading of this disease further. This is the typical symptom, grapevine fan leaf like appearance, fan leaf disease appearance. And this is the dagger nematode, Ziphinema index or Ziphinema italiae, which are responsible for transmitting this disease. Besides grafting and budding from the infected plants, the infected plants are having sharp edges and asymmetrical in nature with chlorotic streaks around the veins. And then second important viral disease is the grapevine leaf roll virus. The name itself indicates the infected leaves turn into reddish brown in color and then roll along the margin downward, so downward rolling of leaves along the margins, and you could see the presence of reddish brown discoloration in the intervenal area due to viral infection, and this is caused by the grapevine leaf roll virus and got transmitted by the mealybug Planococcus ficus, and the leaf tissue between the veins turn into deep red or purple, and downward cupping and curling of the leaf along the margin are very common. On the white varieties, the leaf tissues will turn into yellow with the curling and cupping of leaf margins. And to manage this disease, removal of affected portions which will reduce the inoculum pressure, and then monitoring of the crop for the occurrence of mealybug is very important, and the soft scales formation should be indication of the mealybug infestation. So when the mealybug infestation that occurs, and quite frequently you could see for the development of this grape leaf roll disease. So better control the mealybug infestation by spraying any of the pesticide effective against the sucking pests. And then Pierce disease of grapevine, which is caused by the fastidious bacteria Xylella fastidiosa, which is a xylem inhabiting bacteria, gram negative bacteria. So the infection by this Xylella fastidiosa, which is causing sudden drying and scalding along the margin of the leaf, while the rest of the area of the leaf will remain still remain green, and they advance more towards the center, and severely affected leaf area become completely dried and fall off. Similar type of necrotic lesions may also be formed on the petiole as well as on the stem, and this pathogen, that is Xylella fastidiosa, which could be transmitted by the sharpshooter Draculocephala minerva and the red-headed sharpshooter Carniocephala fulgida. And the pathogen responsible for this Pierce disease is Xylella fastidiosa, which is a gram-negative xylem limited bacteria, slow growing bacteria, and got transmitted by the xylem feeding insect vectors mentioned above, green sharpshooter and red-headed sharpshooter. And the main management strategy recommended for the management of this disease is the removal of affected plant portions to avoid further spread or to avoid the inoculum density, and controlling of the insect vectors by application of any of the effective insecticides.